embryonic layers and coelom. Diploblastic organization. In diploblastic animals, cells are arranged in two embryonic layers, ectoderm and endoderm. Ectoderm gives rise to epidermis, which forms the body wall. Endodermis gives rise to gastrodermis, which forms the lining of the gut cavity. Mesoglia is undifferentiated and present between both the layers. Example: Cnidarians and ctenophore. Triploblastic organization. These animals have three layers: ectoderm, mesoderm, and endoderm. They exhibit organ system level of organization. Organs are formed by these tissues. These organs form excretory, nervous, digestive, reproductive, and circulatory systems. Triploblastic animals are bilaterally symmetrical organisms. Coelom. The term coelom was coined by Haeckel. Coelom is a fluid-filled cavity that is developed within the mesoderm of triploblastic animals. The fluid-filled body cavity protects visceral organs by acting as a shock absorber. Early stages of development. The zygote of triploblastic animals undergoes a series of mitotic divisions called cleavage. These cells are called as blastomeres. The cleavage proceeds further and forms a hollow ball of cells called blastula. The central cavity of blastula, called blastocele, invaginates at one end and forms the gastrula. Ectoderm is the outer layer of gastrula, the endoderm is the inner layer, and blastopore is the opening. The cavity between ectoderm and endoderm is known as the blastocele. The layer which is developed between embryonic ectoderm and endoderm is called mesoderm. Based on the presence of coelom, organisms can be classified as acoelomates, pseudocoelomates, and coelomates.